Um, Thank you. Thanks for the introduction, Pai Reinhardt. I'm Yue from University of Pittsburgh, and it is my great pleasure to have this opportunity to present our study, Perturbed Transcriptomic Analysis, identify chemo-immunotherapy synergisms to shift anti-PD-1 resistance in cancer. Here's my disclosure. So the anti-PD-1 therapy is one of the most successful immunotherapies. It can block the inhibitory checkpoints and restore the host immune system to recognize and kill the tumor. The immunotherapy has brought a revolution in cancer treatment, making a lot of patients with malignant cancers and those who failed to respond to the chemotherapy survived with better living quality. But unfortunately, the overall response rate in the entire cancer patient population is still low, and this is a big problem of the immunotherapy. So to let more patients benefit from the immunotherapy, FDA has approved several chemo-immunotherapy combination regimens. For many standard of care chemotherapies can induce immune response and sensitize the tumors to the immunotherapy. But the thing is, the current chemoimmunotherapy combination regimens are still within limited cancer types and drugs, and we still have a long way to go. So what we're doing now is trying to systematically identify the chemotherapy agents that can be used with the immunotherapy to reverse the immunotherapy resistance. And we proposed a novel integrated analysis method called Perturbed, which is based on the post-treatment transcriptomes to predict the synergism between the chemotherapy and immunotherapy. So the reason we work on the perturbed transcriptome is that we believe the transcriptome profiling after the drug treatment will let us know what is exactly happening in cells after because of the drug treatment. So in the Perturbed, we start from a patient data with paired expression sequenced before and after the patients are receiving the immunotherapy. And we are trying to look for the exact feature that is make a patient resistant to the immunotherapy. And then we utilize the transcriptome of data of the cancer cell lines after the shRNA perturbation from the connectivity map. It's a great database to investigating what would happen if the features related to the immunotherapy resistance are being suppressed in the cancer cells. So patient data comes from a published study in 2017, which includes the transcriptomes of 33 melanoma patients opting before and during their anti-PD-1 therapy. Since now we have gene expression before treatment, gene expression during the treatment, we are curious about which of them can best differentiate the responders and non-responders. So we use the gene expression before treatment, gene expression on the treatment, and also the treatment-induced gene expression changes, which is named the tag here, to classify the patients to predict the anti-PD-1 response. And we found actually it's not the expression before the treatment. It's also not the expression during the treatment, but it's the treatment-induced gene expression changes. It's the tag that achieve the best performance in predicting the anti-PD-1 response. So we conclude that the tag is the predominant feature that can explain the difference between the responders and non-responders. So what we're doing next is to establish the gene signatures for anti-PD-1 responders and non-responders based on the most differential tags between them. And we found the genes in tag signature for the responders are enriched in the immune relevant pathways like interferon signaling, the inflammatories. But in contrast, the genes involved in the tag signature for the non-responders are more enriched in those cancer hallmarks like the MYC targets, the cell cycles as I highlighted here. So for easier interpretation, we named these two gene signatures as tag for sensitive signature and also the tag for resistant signature. So we also look into the tumor samples from the TCGA, and we found a higher expression of TAC4 sensitive signature is correlated with higher immune cell infiltration, whereas the higher TAC4 resistant expression is correlated with immune cell exclusion. And we also observed that the genes in the TAC4 resistant signature are actually associated with worse prognosis, whereas you can see the TAC4 sensitive is actually going to the opposite direction. So the take-home message here is that we discovered that the TAC signatures are determining whether a patient will respond to the immunotherapy. So our next question is, what will happen to the tumor if we inhibit the TAC4 resistant genes? Will the tumor become sensitive to the immunotherapy? So to answer this question, we collect the transcriptome of cancer cell lines after shRNA treatment from connectivity map database. And these shRNAs are targeting the genes from TAC4 resistant, and we use the gene expression change intensity score to measure the influence on the TAC signatures after the shRNA treatment. So surprisingly, we found that if the genes from the TAC resistant signatures are being knocked down, there will be an overall expression upregulation of genes from TAC4 sensitive. And if a TAC4 sensitive gene is being suppressed, the cell will express more TAC4 resistant genes. So these results are telling us that actually these two TAC signatures are not just signatures. Instead, suppressing of the TAC4 resistant can make the cell expression in a way more like an immunotherapy responder, which means a more active immune response and a more sensitive state to the immunotherapy. So, therefore, we developed a matrix termed shiftability to identify the treatment that can suppress the TAC4 resistant and promote the TAC4 sensitive. And here shows the shiftability of shRNAs targeting the TAC signature genes in a melanoma cell line, and we can see the shRNAs targeting the TAC resistant genes are shifting the cells to the to an anti-PD-1 responder phenotype, and shRNAs targeting the TAC sensitive genes are actually going to the opposite direction. So based on this shift theory, we carry out an in silico screening on 1 million compound-treated transcriptomes from the connectivity map database. We start to search for the compounds who can induce such shifting and sensitize the cells to the immunotherapy. So in total, we identified several hundreds of drugs who can induce the significant shifting to the responders in at least one cell lines. And here I listed the ones who have shown significant shifting across multiple cell lines. And I put the highlighter here. So these are the ones, are the drugs and compounds who have already been reported to have synergism with anti-PD-1 or have something to do with the immune response induction. It's a very nice recapitulation of the reported cases of chemo-immunotherapy combinations. And these include those MTX and doxorubicin. So those are typo isomerase inhibitors, and we also have HDAC inhibitors, CDK inhibitors, and some ribonucleotide reductase inhibitors. So, but we are not stopping at this point. So besides those reported cases, we also want to characterize a short list of potent chemoimmunotherapy synergy targets. So in the future, we can do some drug design and drug screening with some more specific focuses. So we integrate the shiftability results from the shRNAs and compounds. And that's actually the beauty of these post-treatment expression profilings, because you can capture the real changes brought by the blockade of a gene through either the genetic inhibition or pharmacological inhibition. So the one I highlighted here is one of the potent synergy targets we identified. It is the PAK4, the P21 activated kinase 4. So we have validated in multiple cell lines that the PLK4 inhibitors can induce a dose-dependent increase of genes involved in antigen presentation, type 1, type 2 interferon signalings, and PD-L1 expression. So, summary. Our method, called Perturbed, proposed that treatment-induced expression changes can serve as a novel molecular phenotype to describe the immunotherapy response. We have highlighted the shifting between them through the introduction of drug treatment. And based on this novel theory, we have provided a reliable and a systematic synergism prediction for immunotherapy and chemotherapy combinations. And if you're interested in the details of our study, please feel free to stop by our poster. Thank you all for listening, and now I'll take any questions. Okay, the question, yeah, I will repeat. So if I understand correctly, so the question is, because the prediction is based on the treatment-induced gene expression changes, so which means you need to take the treatment and know the expression changes because of the treatment, and then you will know whether the patient is responding to the treatment or not. So if that is kind of like contradictory. So answer is, so the key point of this study is not actually predicting a patient response. So it's more than like prediction. It's like we are trying to figure out the drivers, the reasons of a patient, why he or she is resistant to the therapy. So it's more like a prospective studies. So after the treatment, you know the changes in the patients, you know the difference between the patients, and you find out the drugs that can reverse these kind of changes and improve the treatment outcome. So yeah, thank you. Thanks for the question. Yeah, it's actually a very good question. So the question is, we get the tech signatures from the melanoma patients, but when we do the cell line screening, we're actually looking for other, so it's not limited to melanoma cell lines. We have multiple cancer cell lines from different cancer types, different subtypes. And how can we make sure that the tumor heterogeneity between the cancers will not affect the results? So the answer is, yes, so one of the limitation I would say of our study is the signature is getting from the melanoma patients. So for sure, these signatures contain some melanoma specific features. But when speaking about like tumor intrinsic mechanism of immune response evasion or immune response induction, is actually they have some common pathways between the cancers, even between different cancers. So what we are trying to do is a kind of a preliminary screening in the cancer cell lines. So we hope to find out those tumor intrinsic immune response induction, the most common ones. And for sure, if we want to apply this study to a more advanced stage, we need to do a lot of like a mouse model, a lot of cell line validation. So I think in that stage, we will address the cancer specific things in a more detailed way. Yeah, Thank you. Thank you for the question. So the question is, do we have already a model? So we have a mouse model ongoing, and we're still trying to optimize the conditions, because it's kind of you apply the chemo drugs and you apply the immunotherapy drugs together into the same system. So it still requires some time to explore the conditions. Yeah, it's a kind of new system. Yeah, thank you.
Very good presentation. I just have a quick question about your drug list. I took a picture so I can zoom in and see.
Yeah.
It seems that a lot of them are MEK inhibitors. Have you tried to look into some public data sets where they have melanoma pre and post-treatment gene expression, just to see that whether patients after receiving the MEK would induce some of your MEK inhibitors?
Okay, so the question is, our top list actually has a lot of MEK inhibitor. So because actually our signatures is from melanoma, and melanoma patients are treated with a lot of MEK inhibitors. So yes, it's actually a good point. So we are looking for some like drug treated data sets before and after you have the gene expression available in the clinic, and we are actually searching for these kind of data sets to see whether those patients treated with these chemotherapy or small molecule inhibitors, they really induce the immune response in the tumor. So yeah, we will spend some time on this part. Thanks for the suggestion.